Hello, everyone. I'm going to officially welcome us to today's Lunch and Learn. Continue to get your lunch if you haven't. We're going to just talk for a few minutes before we introduce our keynote speakers. So don't rush, take your time, and enjoy lunch. Welcome to today's Lunch and Learn. My name is Sarah Imbro. I'm the Executive Director of ALS United of Georgia, and we are just so pleased to have you all here today. Thank you for making the effort. Want to give a little bit of recognition to who has helped make this day possible. So if you'll just bear with me. Hopefully you've seen some of our partners today. Our premier partner sponsor is, we lovingly call them MT Pharma because it's a long name to say otherwise. Our gold sponsor partners behind us, our silver sponsors, and our bronze sponsor partners. So these partners, a lot of them are here today with you. Hopefully you saw them, had time to see and speak with them in the adjacent room. And they'll be joining us for lunch as well, so maybe sitting at your table if you have an open seat. Also wanted to let you all know a little bit about one of our programs. Some of you all have already participated in this program before, but our Caustic Care Grant program is beginning its second grant cycle. It does open on Monday, August 4th. We do not accept applications early, but you can go ahead and get your application filled out either via hard copy today at our care services table or through this QR code on our website. For those of you that are not aware of this program, this is a $1,000 grant reimbursable program, and it is designed to help you with needs that are typically not reimbursed by insurance, Medicare, or Medicaid needs. And there is a whole laundry list of things that you could use this for, so we encourage you to take a look at that information and see if that's something that could be a benefit for you when this grant cycle opens next week. Before we go any further, I would be remiss if I did not take a moment to introduce our team. So for those of you that have been here for a little bit this morning, many of us are in this light color blue, whether it's logoed brand clothing or just the color blue, and that is our staff team here at ALS United of Georgia. I am so fortunate to partner with so many great people. I want to take a minute and recognize them, and they're kind of scattered throughout the room, so hopefully I won't miss anyone, but you can raise your hand. But we have incredible care services team. Many of you know the names, but maybe not the faces, so today you get to see their faces. So we have Anita Flowers over here, our senior program manager. We have Robin Rakoff and Stephanie Medill over here with our care services team. If you attend multidisciplinary clinic, you typically see them on site at clinic and email and talk to them a lot. Our administrative coordinator who keeps us all in line and things running efficiently on the back end is Pam Amy Cup back here. You saw her at the registration table. And then we have our development team as well, led by Liza Nornmark, our Senior Director of Philanthropy. You're going to hear from her in a minute. Kelsey Thomas over here, our Senior Events Coordinator. And we have Ellen Strickland, our Donor Relations Manager. And newest member to our team is Allison Bailey, and she is over there as well. So we're just so grateful to have a team that can help you on your journey and answer your questions and just really be a guiding light for you in answering those questions and pointing you to resources. If we don't have it and can't provide it, we want to be able to help you find a way to get what you need, and that's what we're here for. I want to invite Liza to come up and talk to you a little bit about some other events and happenings, and then we will introduce our keynote speakers. That's just a move. Just hit that clicker.
Do I need that?
No, I can do it.
Okay, you do that. Good morning to everybody. Thank you so much for being here. It's wonderful to see some familiar faces and meet some new people. So Sarah, button. We're so glad that you're here today and part of our community, and we just want to formally invite you and make sure that you've heard about the walk that is coming up on September 20th. This is the 24th year that we've had this event, which is amazing. So, yes, it is such a wonderful day. It is a day of community. We joke about it, but it's true. We call it our Super Bowl. And it really is. It's just the day that we all come together and celebrate everyone who is living with ALS, everyone who has, loved somebody on a journey with ALS. It's truly, truly a special day. So make sure you visit the girls over at the walk table. We would love to tell you more and register you. The walk is so important because the funds that we raise from the walk go to support our programs. And we have more information about that over here with our care services team. But programs like the Sally Panfell in-home care and respite program, the Caustic Care Grant program, like Sarah just mentioned. We have the Paul B. Williams Transportation Program, which I know many of you have used. We have our support groups, which are in person and virtual, equipment loan, our amazing care services team. The funds also help us support the clinic at Emory and research that's happening across the country. So it's really, really important and just such a special day, and we would love for you to come out and join us. Again, if you have any information, if you want information, we have it, and we'll be over at the walk table. So we look forward to seeing you there. And now we're going to bring Sarah back up, and she's going to introduce the doctors.
So the main reason you all are here, not to see me or Liza, but to see these two individuals that we're bringing on the stage here. Many of you know Dr. Glass and Dr. Fournier from the Emory ALS Center, but just a little bit about their work is the Emory ALS Center began 28 years ago in 1997 with Dr. Glass leading the charge and still doing so. It's his vision to be able to help people with ALS, and I'm sure he will tell you a little bit more about that. With his co-director of Christina Fournier, she has been, how long have you been with the clinic now? 13 years, as long as I've been with ALS United. She is also the director of ALS care at the Atlanta VA as well. So if you're a veteran and received care there, you know her from that clinic also. Many of you know the Emory ALS Center is a nationally recognized clinic center for ALS. It provides comprehensive care for people and families living with ALS. And in addition, Emory is focused on ALS research, both in basic science and clinically. So they're pursuing the answers to why ALS happens and what makes it progress. And yes, that is on their website. So if you haven't taken a look at their website, it's there. So they are going to talk to you today about a little bit about their clinic and about ALS research, what they're involved in, and what they're seeing on the landscape, and how maybe you can participate at some point in time. We did invite questions. They are going to do a Q&A panel as well. So if you have not already, there are note cards or paper on your table with some pens. We're going to have you fill out those questions. If you have a question during the talk, just raise your card up and members of our team will grab them, and we'll try to get to as many as possible. And we have a good few to start with if you sent any in via email or before the program started this morning. But we will do our best to get through as many questions as possible. So at this point, I'm going to turn it over to Dr. Glass and Dr. Fournier, and I will help read questions when you're at that portion.
Good morning, afternoon, everybody. Some of you know me, some of you don't. I'm Dr. Glass, but I'm going to turn this over to Dr. Fournier to start. What we're going to do here is give you just a little smidgen of what we're doing from a research standpoint, because I think that may be what most of you want to hear about. I think many of you know what ALS is better than we do, to tell you the truth, if you're living with it or you're living with somebody with it. So we're going to tell you what we do at the clinic, what's important to us, and what's important to you, we hope, and then we're going to spend a lot of time answering your questions. So I'm going to turn it over to Christina, and you go.
All right, thank you. It's nice to be here, nice to see a lot of familiar faces. So we did want to leave most of the time open for questions, but we thought we would explain a little more about what we call, for short, the CRYAL study, which if you've been to our clinic and given your blood and your information and questionnaires and some people even spinal fluid and skin, you're helping us to learn more about ALS and researchers all over the world to learn more about ALS. And we thought we'd give a little context about what is this study and what are you actually a part of when you participate in the clinical research and ALS study. And so really the goal is that every person who comes to our clinic teaches us more about ALS, that you're contributing to finding the treatment for this and that we learn more from you. And so we do that by collecting both clinical information and looking at kind of your biologic samples and tying the two together. And that really gives us an important piece of the puzzle, particularly because, as you could see today, like everybody's ALS looks a little bit different, right? It starts in a different part of the body, different ages, different backgrounds. It looks differently, and we need to understand why that is to develop treatments and potentially to come up with the right treatments for different types of ALS. And so that's what this is all about. And so what you guys have helped us build so far is we have more than 2,000 people with ALS that have participated in the study and been generous with their information and their biofluids, and that we've tracked over time. And this really helps us to understand kind of why ALS looks different and what's driving it. We collect the data at every time you're at the clinic with the questionnaires and with the exams and with the samples that we get. Have more than 1,800 blood samples, spinal fluid. Even some people donate the autopsy after they pass, and we have all of that, and it's all tied together. So it's not just these little samples in isolation, but we have kind of the whole clinical course tracked and linked with these biofluids over time. And this is such a valuable resource that's going to help us to kind of come up with meaningful treatments. So it's very generous that so many of our patients agree to do this. So I was going to talk a little bit about kind of what we can learn clinically from this, and then I'm going to hand it off to Dr. Glass to talk more about some of the things going on in the lab. So I have a personal interest in kind of outcome measures, meaning how we track ALS, and it's not the sexiest story, so I'm going to try to tell you why it's important. And what you see here is if you've ever been given the ALS functional rating scale, this is a questionnaire that's used all over to kind of track ALS progression. It asks about your speech and your swallowing and using your arms and legs and breathing, and it's scored anywhere from 0 to 48, and this is tracked at each visit, and it's used for many of our clinical trials to kind of track how people are doing, and used as the outcome to see, like, is this drug that we're testing working? And in this graph here, this is a sample of people coming into our clinic and what their ALS FRS is doing over time. Each little colored line is kind of the trajectory of an individual patient. And what you could see is it's really wildly variable, right? There's people that progress very quickly. There's people that progress very slowly. There's some people that bounce around a little bit. And so if this is kind of the problem, is we say are testing a drug that we hope slows progression of disease, and maybe it doesn't work the same in everybody. And if this is the backdrop on which you're trying to measure this kind of modest change, it's really tricky, right? So it turns out that how you measure clinical change and progression is really important, because if you want to see a treatment signal through kind of all this noise, then the way that you track progression ends up being super important. So when you humor us with all these different questionnaires and exams and things, a lot of what we're doing is trying to figure out how can we track progression more accurately and kind of see a signal through the noise. And so that's why kind of giving some thought to how we track this ends up being so important. And so one thing we developed at Emory is a questionnaire that was new several years ago called the ROADS, and you've also probably done this in our clinic at your visits. But what this does is it's also a clinical outcome scale that's designed to be patient-reported, meaning that you, the person with ALS, is the one giving the answers about different functional tasks, and it measures change. But it kind of fixed some of the problems mathematically with the ALS FRS, which we expect to be able to track progression more accurately. So I swear I'm not going to give you too much math on a Saturday, but let's see, do I have a laser pointer? Bottom. Okay. So with the ALS functional rating scale that we've been using, I think since the 1990s, each point on that scale, like sometimes it represents this really big functional change, and sometimes it represents this little functional change. Like 1 point doesn't really mean anything, which if you think about we're using this total score to track your progression, that ends up being a problem because sometimes drugs are approved on like a 2 or a 3 point change. And so you really need like 1 point to mean 1 unit of change. And so the roads kind of mathematically fixes that, where like 1 point on the roads is a mathematical measurement unit of change. So for everyone here who did these roads questionnaires and does these roads questionnaires, you're actually giving us a better way to track ALS, and now it's being used in clinical trials and in the clinic. And so we're hoping that this would be kind of a more sensitive measure to track change. Also, we get asked a lot with certain treatments that might be modest, like, is this treatment important? And no one had really measured this. And I think, so if you're gonna take a drug and maybe it has side effects and maybe its effects are modest, like we need to understand what's a clinically meaningful change, right? And that's not gonna be I shouldn't decide that, you guys should decide that, right? Because you're the ones with ALS. And so we've also used all of these questionnaires and things to have patients tell us kind of what amount of functional change matters to you. Like, what do you consider clinically significant? And with what we've captured in the clinic, we've been able to kind of publish some work on what do patients report as clinically meaningful to help us, your clinicians, tell you, well, on average, patients report that this amount of change is meaningful. So this research, I think, is really important because when we do any kind of clinical trial, it puts any changes in context of what does someone living with ALS find important. And so that's another thing that just by answering all these questions, like we've been able to learn from your participation in our study. Another thing that is in the works, and it's actually open for, there's a group called Everything ALS that has an app and does their own digital tracking study, and we collaborate with them. And we know that motor movement is such an important piece of ALS, and this is an app, it kind of takes you through a variety of motor tasks, and it'll describe the task, and it will also do a demonstration of the task, and you kind of rate your own motor performance. And it's a way that either you could track your own progression over time or we, the clinicians, can track progression. But this is cool because it tracks the actual motor movement, and you can do it yourself on the phone without the clinician having to guide you through the exam. So we think this is another way that we can better track progression and track changes over time and track treatment responses by actually looking at the motor movement, tracking it over time. And we know coming to the clinic is challenging, so this gives a way for exams to be tracked without you having to come in. And then everything that we do clinically is correlated with these blood samples, with these biomarkers. We know ALS isn't simple, right? We don't have just it's not like diabetes where you just check a blood sugar and we know how you're doing. We're trying to figure out markers in the blood and markers that predict how your ALS is gonna behave or how your ALS may respond to treatment. And so all of these clinical things I talked about, we're correlating with the biospecimens, and I'm gonna pass it off to Dr. Glass to kind of tell you the lab piece of this and what we're doing there.
Thank you, Christina. I'm actually going to stand up so I can see what I'm looking at. So I just got to tell you, everything that Dr. Fournier has just told you is extraordinarily important. Because if we're trying to figure out whether a drug works or not, it's very important to be able to measure very specifically how patients are progressing with their disease. And if we don't have a very sensitive way of doing that, we may make mistakes. And I think the clinically meaningful issue is very important as well. So yeah, we can get all kinds of kudos and great publications and progression through our academic careers by doing this kind of stuff. But also the clinical stuff, working with people who actually have the disease, which I'm going to tell you, Dr. Fournier is the one who's really driving all this. My name may be on those papers, but she's driving this truck. So, and I can't tell you how much I appreciate that. The other thing I'm going to say is that, before I tell you what's up here, is that I've done this for a very long time, for over 30 years. And I used to spend a lot of time using animal models of disease, which I think are very helpful, and spent years and years and years doing experiments on laboratory animals. But I'm not sure that it taught me as much as dealing with people with this disease. In fact, I would argue that the most important experimental model for ALS is people, because nobody else gets ALS but people. That's it. And so I have really changed my research focus toward studying people. And when you come to the clinic and you give us blood or spinal fluid or skin, where I can grow your cells and culture, I'm actually studying people. And I think that's a much more relevant thing to do these days if we're really trying to understand what causes this disease. With at least my philosophical idea is that if you don't understand what causes a disease, it's very, very difficult to find a treatment for it. And as Dr. Fournier has already shown you, this is such a variable disease. In fact, look around the room, it's such a variable disease. We don't even know it's one disease. It may be multiple diseases, and we need to figure that out, because if we're going to figure out how to treat this disease or these diseases, we got to know what type of disease you have, because maybe that one drug won't fit everybody. Maybe it's just for this group or this group or this group. And that introduces what we're doing here. And so this is a paper that we published earlier this year. And it's got a long title, but basically this is a technique that we've really is our basic technique in the laboratory now, which is called proteomics. Proteomics is basically the study of proteins. Everything that you are is a protein. And so your DNA, which you're born with, gets transcribed into something called RNA, and RNA gets translated into proteins. And everything we are is proteins, okay? And so we've been studying proteins, trying to figure out what differences there may be in people who have ALS, different types of ALS, or even compared to controls. So in this study, you can see here, does this work? This one? Oh, okay. It just doesn't show up very well. Oh, there it is. It's up here. It's not there. Okay, on the left, what you can see is the number of patients that we started with, okay? Controls, meaning people who don't have disease, okay? People who are carrying the most common mutation that causes this disease, which is called C9orf72. It's a mutation that we see very regularly in the clinic. We have 59 people who are carrying the mutation but don't have the disease, meaning that they're at risk for the disease, and they may develop this disease later on, but right now they're asymptomatic, okay? We actually have people who actually have the disease, C9orf72, 43 of those. And then there's another relatively common mutation called SOD1. That's the one that was discovered first back in 1993. And again, we have some asymptomatic people carrying that mutation. Then we have people who actually have ALS with that mutation. And then we have people who don't have genetic ALS. We call those sporadic ALS or SALS. And that's the majority of people. 9 out of 10 people with ALS have sporadic disease. And what we do is we take their spinal fluid, at least in this case, we took their spinal fluids and actually digested their proteins, put them through a machine called a mass spectrometer, and then can look at things that are different between these different groups. And here, what you're showing here, and this is just a small piece of this publication, what you're seeing here is this, on the left, the sporadic ALS versus the control group. And on the right, you're looking at the people with the C9orf72 ALS versus the control group. Now we have these kind of things. And for obvious reasons, these things are called volcano plots, right? Because this looks like it's a volcano, right? And basically everything on the right in red means that in patients with sporadic ALS, at least in the left graph, those proteins are higher in their spinal fluid than in controls. And the blue ones on the left are the ones that are lower. So we can see right then and there that ALS is different than control in the spinal fluid. the proteins are different. We can also see that if we compare just the C9orf72 patients to the controls, we also see differences. Some are increased, some are decreased. But in fact, I don't have this up here, but if you just compare the C9orf72 ALS to the C9orf72 asymptomatic carriers, they're also different. Now, why is that important? Because we don't want to just treat this disease, we want to prevent this disease. So if we can see a protein pattern that changes just before people get sick, and if we had a drug to treat this disease, we might be able to start it before they ever get sick. And so that's really the holy grail here, is to try to figure these proteins out and see if we can find what are called biomarkers that actually tell us, aha, this person's about to get sick. We've now moved on from mass spectroscopy, which is that machine I showed you there, to a very new technique that's called NULISA, or nucleic acid-linked immunosandwich assay. It's a lot of words. But basically, instead of putting this through this mass spectrometer, which basically takes this gamish of proteins, grinds them up, and puts them through a machine and spits out what you have, okay? This is actually what's called an antibody-based system, where we actually can make other proteins, which are called antibodies, that attach to proteins. And when they attach to those proteins, we can then capture them and then look with molecular techniques to see how much of that protein is there. So there's an old thing that we've done in the laboratory for decades called ELISA, or enzyme-linked immunoabsorbent assay. Now this is called NULISA, okay? Where basically we're using nucleic acids or DNA to actually target these things. And this is a much more sensitive and much more quantitative technique. That's kind of an overview, and if anybody's really interested in this, I can tell you later. But what we've been able to do then, and this is a paper by one of our junior colleagues, Eleanor Thomas, who's now working in the laboratory and also is an ALS clinician that some of you may have seen in the clinic. What we've now been able to do is to look again, this is again, control, asymptomatic C9 ALS versus ALS, and we now can quantify those proteins in a different way to see what the differences are. And if you look at the top left, it says NEFL. NEFL is also called neurofilament light. Some of you may have had neurofilament light tested, and in fact, we're doing this now clinically in the clinic. But what you can see is that people with ALS have a much higher level of this neurofilament than the controls or the asymptomatic people. Why is that important? Well, people with asymptomatic C9, they are at risk for getting ALS. But this biomarker of neurofilament tells us that they don't yet have ALS. And when that neurofilament goes up, they probably do have ALS. What we're trying to do now is to say, okay, are there other proteins other than neurofilament that may move quicker than the neurofilament than NFL to identify people who are at risk for actually going from asymptomatic to symptomatic. The other thing we know about NFL is that the higher your NFL level is, probably the faster your disease is going to progress. And so that's very important for us to try to understand how people are progressing with disease. And in fact, what we're working on now in the laboratory actively as of this morning, today, sitting in my office working on this, is developing predictive models using these kinds of measures, whether it be the neurofilament or some of the other major proteins that we're looking at here, including TDP43, which is on the bottom right, which is the protein that we're very interested in patients pathologically with ALS, that if we can combine all these things, maybe we can actually develop a model that will predict how people are going to progress, which is going to be very important, not just for individuals who want to know that answer. I get that question every day. But it's also important for clinical trials to try to understand if we can get a group of people that are progressing at the same rate, maybe we can actually be better at testing some of these drugs. Another thing that we have done, how many of you know about the C9orf72 antisense oligonucleotide clinical trial? Okay. How many of you know about tofersen and the SOD1 antisense oligonucleotide joint? Okay. All right. So antisense oligonucleotides are really the latest and greatest approach we have to treating people with genetically based disease. And in fact, something that has proved to me and has changed the way I think about this disease is the development of a drug called tofersen, or now it's called Qalsody. Thank you. I don't know where they get these names from. Okay, and that's a drug to treat people with SOD-related ALS. I showed you some people in our research program. It is an amazing story in that what this antisense oligonucleotide does is it goes in, you put it into the spinal fluid, and it's a piece of RNA and DNA that bind to the gene that makes SOD1 and actually turns it off. And when it turns it off, at least in people with this type of ALS, SOD1-related ALS, okay, it actually stops, in some people, not everybody, it can stop the disease from progressing, and there are some reports around the world of people starting to get better. So ALS is now a treatable disease. That's the message here. ALS is a treatable disease, at least in this very small population of patients. But it means we have our foot in the door, and maybe we can now expand that to other people. And that's happening now with people with other genetic mutations. So we tried this with the most common mutation, which is called C9orf72. And we had a lot of high hopes for this one. We had another antisense oligonucleotide that was going to bind to that gene and turn that gene off. It didn't work. And in fact, that trial, it was tried by several companies, by the way, and all those trials failed. And the real question was, why did they fail? Now, in reality, because of a lot of what we have from our CRIALS program, including DNA, spinal fluid, blood, and autopsy tissue, we were able to start to ask that question. And here's a paper that's now in press, where we actually looked at the neuropathology of patients who were in this trial and died. Where's the drug? Where did it go? Did it go where it was supposed to go? Okay. What did it do? And so we use our neuropathology as well as our proteomics to actually answer some of those questions. I'm not sure if I have, yeah, okay. And so the top is just, these are just two ways of looking at it. The top is called immunohistochemistry, which is an antibody-based way of looking at it. And you can see the bottom is a much more sensitive way of looking at the drug. All the red is drug, okay, on the bottom, in the bottom here. We'll ignore the top for now. Everything that's red is drug, which means that drug is there. It's in the spinal cord, but in fact, I don't have these other pictures, but it's in the brain as well. And the drug actually sticks around for more than a year in patients. So we answered that first question. Did the drug get there? This is the first time this has ever been asked, by the way. Yes, it did, okay? What did it do, okay? Well, we looked at the pathological changes that we associate with ALS, and we looked at those kinds of proteomic changes that I showed you were associated with ALS, and it didn't do what it was supposed to do. So now we have part of the answer of why it didn't work. It got there, but it didn't change the pathology, and it didn't change the proteomics. But what we have found is that it actually created some changes that we're now pursuing that may actually have suggested why it didn't work. Not necessarily because it didn't do what it was supposed to do, but maybe it did something bad. We don't know yet, okay? And so my point here is that when you donate your blood, or your DNA, your spinal fluid, or unfortunately, postmortem tissue, it is used for these kinds of projects where we're going to figure out, number one, what this disease is in terms of how many different types of diseases there are. Number two, how it works. And number three, potentially how to treat it. Okay? So I don't think I have anything. Oh, sorry. It's in the rest of the brain as well. I already told you that. Here it is up in the cortex and the cerebellum. And you can see all the red, that's all drug. So it got there. Oh, okay. I didn't know I had this in here. Thank you. So in fact, in this same project, we actually had spinal fluid from these patients as they were being treated during the course of their disease, because all of this drug gets put in through the spinal fluid, okay? And when we do a spinal tap and we give the drug through spinal fluid, we actually take a little bit of that spinal fluid first and put it in the freezer so we can do research on it later. And this is what happens. And what you're seeing here, this is our proteomics that I showed you before, top right, okay? Here's a patient that is on the placebo, not on the drug, okay? And again, those of you who have been in drug trials, you usually have a group that is on the drug and a group that's not on the drug so we can compare what happens. So there were people who were not on the drug for a while, but then later on, they get put on the drug. That's called the open label extension, okay, when everybody gets the drug. And what you're seeing here is that during that placebo phase, at least in this group of patients who are on placebo, these proteins that I'm measuring didn't change at all. But as soon as we gave the drug, these proteins, the drug is called BIB-078, by the way, boom, it went up. What are these proteins? What are they doing? And are they important in terms of telling us, understanding why this drug didn't work in these patients? So again, the collection of all of this human material, whether it be clinical data, whether it be the roads and the FRS and the motor exam and all those things associated with spinal fluid, DNA, and autopsy tissue, allows us to put the whole thing together. And when we put the whole thing together, what we hope to do is to actually figure out what causes this disease. And once we figure out what causes this disease, I think we'll be well on our way to figure out how to treat it. So I think hopefully that's the end. Yes, that's the end. Okay. So I think we're going to do some questions and answers. Is that right, Liza?
Okay. Ooh, can you hear? All right. Okay, the first question is, what are the most promising clinical trials going on currently? And where can people find information on clinical trials that are available?
So I have to give the caveat that I think if we knew which clinical trial was the most likely to work, we wouldn't do clinical trials, right? So, and I think we've both been burned on putting our eggs in a basket and having that be the wrong answer. So I think that the short answer is anytime you consider a clinical trial, we can certainly talk to you more about the science and why we're excited about it and what are the pros and cons, but we'll never really know if that trial is going to work or not. In terms of where to find information about trials, so the trials that are happening at Emory, we do keep updated on our website at the Emory ALS Center website. And then there's a website called clinicaltrials.gov, where any clinical trial in the United States is posted, and you can search by ALS, you can search by region, and you can look there. There's also another tool, ALS TDI has a clinical trial tracker that I think if you just Google ALS TDI tracker, and I think that they get their information from clinicaltrials.gov, but it's a bit more accessible and user-friendly, so that's another good resource to kind of see what trials are out there. And NEALS, which is the Northeast ALS Consortium, I think keeps trials there. So all of those are good places to go. But I wouldn't pretend to know which one to bet on. But I will say there's the clinical trial landscape, I think, is becoming more exciting in that we're seeing more trials that are targeted towards like ALS-specific mechanisms, or can you manipulate specific ALS genes? And so I think that we're hopeful that we're going to have more successes, but I wouldn't pretend to know which one is the most promising at this stage.
Right. And so a follow-up question is, how do you become part of a drug trial? Is it just going to those websites and applying, or is there another easier way to navigate that?
Yeah, so I'll comment on this. What you'll find is that not all drug trials are being done everywhere. And there's several reasons for that. Many of the drug trials that are run by large pharmaceutical companies, they choose sites that they want to use based on whatever method they use to choose that. And they only usually choose a certain number of places. So not all trials are happening at all places, number one. Number two, at least at our center, what Dr. Fournier and I do is we look at the science behind the trial, and we look at the safety of the trial, all kinds of things, before we decide to sign on and say, yeah, this is good for our patients. Not everybody has the same attitudes towards that, and it may be happening some places and not others. And so what you need to do if you want to be in a trial is find the trial that interests you, and a trial that interests you should not just be, this is a trial. It's how much effort does it take? What's the risks and benefits of it? How do I want to spend my time? How much time do I want to be there? Do I want to have spinal taps or don't I want to have spinal taps? You know, there are all kinds of things that are different in different trials. And so you have to decide for yourself whether you're interested in it, and then you have to see whether you are eligible, and there are certain eligibility criteria. And that's important. Not everybody's going to be eligible to be in a clinical trial. And usually it has to do with how your disease has progressed, how far along in your disease you are, how your breathing is, all kinds of stuff like that. So there's no easy answer to this. But if you're interested in it, talk to the folks who are running that trial, whether it be us or someplace else, and they can give you more information.
I'll add that on those websites, we geared you toward the clinicaltrials.gov, and the tracker for each trial, there's typically a contact person listed, so it'll tell you who to contact if you're interested in that specific trial.
Yeah. So that's a little bit about clinical drug trials. This question is, how many research studies is Emory involved in regarding ALS?
Several. We do clinical drug trials, but we also, this clinical research in ALS, that's research as far as I'm concerned. It may not be what we call interventional research. It's not a drug trial to change the course of disease. It's a trial to understand disease. Christina, you can tell us what we actually have going right now and what's the plan for next?
Yeah, I would say at any given point, we have several clinical trials, which are drug trials, and then several what I would call observational or non-interventional studies, which is we learn more about ALS from people participating, but they're not given a drug. And so, and it's kind of a dynamic thing because there's always trials that are starting and stopping. But I'd say at any given point, there's usually multiple interventional and observational studies. And I think we have several of both currently.
Okay. Talking a little bit about the ALS FRS scale, or ALS functional rating scale, why is this still used to evaluate clinical trials, and what is your take on this? Which you touched a little bit on a new method.
Yeah, so the short answer is, so I think one, on face value, it does ask about things we care about in ALS, right? So the speech, the swallowing, the moving your arms and legs, like those are things that we should measure in ALS. But the short answer is because when tofersen and edaravone were, and Amylyx, which is now not approved, when these drugs were approved, the ALS FRS was used as the way to track progression. And so we know that the FDA and like the European agency that approved drugs accepts this outcome measure as a way to approve drugs. So I think drug companies say, okay, we saw that this is something FDA uses, we will continue to use it. It doesn't mean that we're not incorporating other outcomes that might be better as well, but I think there's this reluctance to get rid of it because a drug has been approved using it. So I think what the compromise that I think we're using now is that, okay, it's the ALS FRS is here to stay and we're gonna do it, but we're seeing novel outcomes in more kind of robust ways of tracking progression also incorporated into trials. So yes, the FRS is used, but we're now seeing these, speech tracking and innovative biomarkers and the roads and things like that are also being used. So I think that yes, it's still there, and there might be some reasons we don't love it, but there's other things that are more robust that are being used as well.
So this is a question about when pharmaceutical companies decide to end a clinical trial for lack of efficacy and/or statistical significance, do they ever consider doing a longitudinal follow-up with patients who were participants?
Yes, they do. In fact, if you look at the tofersen study, which again is a remarkable success in the ALS field, the first look at the data was at 24 weeks, 6 months basically. And it did not reach its endpoint and was not approved based on that. But when they followed those patients out through a full year, 52 weeks, that's when those scales and those graphs started to really separate. And that's when the FDA looked at that and said, whoa, this really does work. And in fact, it's probably taught us a lesson that maybe a 6-month trial may not be long enough to really decide whether a drug works or not. So I think we learned that from there. But there are trials that have stopped, and the question is, once a trial stops, technically, you're not supposed to collect any more data on those patients, because the Human Investigations Committee and the Informed Consent say you are part of a research study until the research study ends. If that research study ends, technically, you're not supposed to actually collect more data. But we do. And we look for changes over time. I don't think, other than that, what I just told you about tofersen, I don't think that has been productive at this point. But we don't forget about patients who have been on trials.
Okay. That was a lot. I can't repeat all of what he said, but he has been a physician who worked with Johnson & Johnson and was making 3 different points. Maybe you heard some of that, but I don't think everyone could hear that.
I think you brought up the issue of compassionate use or expanded access, which allows patients, even in closed trial, but that trial, those patients still need to be part. That can't happen unless there's an ongoing trial also going on. So it's a little bit complicated. But you're right, there are expanded access trials.
Yeah. Okay, thank you. Are there ways AI is assisting your research?
Yes. So there's a lot of interest in this for obvious reasons, but I think AI is really appealing in a disease like ALS where we don't think there's one cause in one measure. And so when we just try to look at one protein in isolation or one clinical scale in isolation, it doesn't tell us the whole picture. But we think that if you can maybe look at a combination of clinical factors and a combination of proteins and maybe some imaging, or maybe you need to put all of those pieces together in a complex way to understand this better. And our human brains are not very good at understanding those complex interplays, but AI is really primed to figure that out. So that's a lot of what, and we're applying the AI to our database, and so all of this data you've given us in a lot of ways, because it's a perfect tool for that, where, hey, we have so much clinical data and so many data points, and we don't actually know which ones are the most important or what combination of things we need to look at. But maybe AI models can tell us, hey, if you look at these 27 data points, you can predict what happens next, or you can predict that, this group of patients actually have shared biology and might benefit from a treatment. So this is an area of a lot of interest in yes, I think that AI is an exciting tool that we're leaning on heavily now for research.
Interesting. Okay, so there are a couple of questions on stem cells. So one says, do you have thoughts on stem cell treatment that you'd like to share? But another one says, are there vitamins or stem cells proven to aid the healing of ALS, and what does research suggest? So if you want to kind of approach the topic of stem cell treatment.
I guess I'm the appropriate one to answer this question. We at Emory was probably the first to actually do stem cell trials in ALS. We did this back in 2009, 2010, 2011. We treated patients with stem cells by injecting those stem cells directly into the spinal cords of patients with ALS. We ended up doing about 30 patients. It was a big deal. In fact, some people thought we were crazy putting needles in people's spinal cord. But this is a desperate disease, and we thought it was a good idea. We had some great science behind it, and we did it. We don't think it actually worked, and we thought that the risk-benefit ratio was not great to move forward. We also ran out of money, but that wasn't the only thing that kept us from doing more. Now, there are stem cell trials going on around the world, but only a few of them are legitimate. I'm going to say that right out loud. The majority of things that you'll find on the internet are not legitimate. They are scams. And they are preying on people who are desperate for some magic that's going to make them better. So be very careful. Be very careful. There is a stem cell trial going on at Cedars-Sinai in California. I think they've stopped recruiting, where they're injecting stem cells. They did a group in the spinal cord, and now they're doing it in the brain, but I think they're closed now, and they've only done about 10 patients. There was a group at the Mayo Clinic who were putting stem cells into the spinal fluid, and I'm not sure if they're recruiting still either. Did not get any great results from that. There was a group in Italy doing this and a group in Korea doing this that were legitimate. Again, did not have any great outcomes from that. So my thoughts on stem cells right now are, I'm not sure that that's the way to go. I think it's been tried in multiple good studies, and that we have not seen any great results from this. And so I do not suggest for my patients to go through that kind of thing. And just another comment about whether it be stem cells or any other, quote, experimental drug, if somebody asks you to pay for it, it's not a real clinical trial, okay? So be careful about that. Vitamins? No. I don't know of any vitamins that actually help with healing at this point. But you should stay well nourished, and if you need a one-a-day vitamin, take it.
Okay. Has any of your research pointed to environmental factors that may cause ALS? From a research perspective.
So there's certainly not a smoking gun environmental factor. Like, we know smoking causes lung cancer, right? There's not anything so compelling in the environment that's a clear contributor to ALS. When you have a disease that we don't know what causes it, it could be that you need a bunch of hits to get ALS. Maybe you have this risk gene and that risk gene and this risk gene and that environmental factor and that. So there could be environmental factors that are like tiny little contributors, but nothing enough where we would recommend a specific lifestyle intervention. I know there's been talk about like golf, I think was the latest one. And and there was some NFL players that got ALS, and we know head trauma is bad for a lot of reasons. It's not super convincing in ALS. So I wouldn't get hit in the head for other reasons. But I think at this point, while there might be multiple tiny contributors to environmental causes, nothing convincing enough where I would say do not do this thing in your environment for the risk of ALS. We just haven't found anything that's compelling. But it is an area of interest, and I know that if it's still funded, the CDC registry has been, tracks like if there's geographic pockets of ALS and if there's environmental concerns. And so it's an area of research, but nothing that's been found that's super convincing at this point.
Right, so to Dr. Fournier's point about the CDC, the National ALS Registry is a survey bank that allows you to provide data on lifestyle and habits and various factors in your life, and they collect all of that data and make it available to researchers should they have a need to find a certain data point. As a follow-up, is there research happening for people living with ALS that have frontal temporal dementia?
Yes, absolutely. We've been doing this for a while. We think that this may be a subset of patients with some kind of different disease. We've actually, years ago, with a student I had, we published a paper looking at the brains of patients either dying with ALS and dementia, or just ALS, or just dementia. And we found that there were major differences in, again, this proteomic thing that we do in those different patients. We also know that there are certain genetic abnormalities that make you more susceptible to dementia. In fact, the most common gene mutation that can cause ALS, which is that C9orf72 I was telling you about earlier, that is also the most common gene mutation that causes familial frontotemporal dementia. And so we have seen families where one patient has ALS, a sibling may have frontotemporal dementia, and then there may be a third person who has both, ALS and FTD. These are very different diseases, as you may expect. In fact, they go to different buildings at our institution. You have dementia, you go here. You have muscle disease or neuromuscular disease, you go here. But they can be caused by the same gene, and in fact, the pathology can be very similar as well. So there is definitely a connection between these two diseases, and it is a very active research topic. In fact, many people who write research grants now don't just talk about ALS, they call about ALS FTD. In fact, the journal is now called ALS FTD, that focuses on ALS, because these two are very well linked. So yes, the answer is yes.
Okay, thank you. So you mentioned NFL in your presentation. What is the significance of NFL levels, and how does it impact research? You might have to define NFL again. Not the football
You don't
league.
need National Football League, right?
It's almost football season.
I saw a guy with the Georgia shirt wander up here. I thought maybe that was it. Yeah, NFL is neurofilament L. Neurofilament is the most abundant protein in neurons and in the wires that connect those neurons to muscles that are called axons. So it's a very abundant protein. And so the idea, although we're not absolutely sure about this, the idea is that when the nervous system starts to break down, whether it be an ALS or multiple sclerosis or head injury or Huntington's disease or other things, when the nerves start to die, they release this neurofilament into the blood and the spinal fluid, and we can then measure it in the blood and the spinal fluid, okay? And so probably ALS is the disease that is most closely linked to the idea that the higher your neurofilament level, the more rapidly your disease will progress. And that data is from our laboratory and many other laboratories around the world. And so it is the one, what we call biomarker, that we really focus on when we're trying to predict how quickly people are going to progress. There are other interesting things about neurofilament. In fact, there's a new data coming out that argues that maybe neurofilament is actually toxic to the nervous system as well when it gets out of the neuron. So we're pursuing that as well. But it is a major focus of all the biomarker work that we do. Did I answer the question?
I think so.
I will caveat here, though, that if you're asking kind of what does my individual neurofilament level mean at any data point, it's just a data point. So when you look at groups of people with ALS, you could see that the group, on average, like a higher neurofilament correlates with a higher progression, but it's not perfect. So if I took your one neurofilament at one time, I can't tell you this is exactly how it's going to progress. It's not that good. It's really more helpful when you're looking groups of patients. So I think that there's a lot of excitement, but it's not so robust that we can just check in one person and that gives you the complete picture.
I think that's important
So I
distinction.
think if someone in the clinic asks, okay, I have this neurofilament level today and this is what it is, what does that mean? Not a whole lot by itself. It's really more helpful as a data point kind of with all the other clinical data. What is the trend and using, looking at it in trials. I think another reason we got so excited about it is in that tofersen trial. So the one drug for that 2% of people with ALS that have the SOD gene, the neurofilament started to drop kind of before we could see the clinical changes. And so it seems to be a really promising marker in trials to kind of indicate that a drug is working. So it's really exciting from a science standpoint, but I think that I don't want to make it sound like we can just check your neurofilament and know exactly what to expect for you. It's not that good, and there is some overlap, and like I said, neurofilament can bounce around a little bit, or it can be high for other reasons. So to you specifically, it might not mean that much in isolation, but for the broader research community, it's exciting.
Okay. You just referenced the SOD1 gene because that is 2% of the population with ALS. How long is the testing process to identify if you have the SOD1? How long is the testing process to identify if you have a SOD gene?
So typically everyone now who gets an ALS diagnosis is offered genetic testing, and that's done through either a blood sample or a saliva sample, and it can take up to a month for that test to come back. And something that we would discuss with a person in clinic about, what are the expected results and what does it mean for your family and that sort of thing. So
Genetic counseling is provided and then an option to do genetic testing? Oh.
I wouldn't give advice to, I would
Understood.
say that if you don't think you've had genetic testing, you should bring that up to your doctor, and it should be offered if you're a person living with ALS. And that's something that could be talked about before the testing is done.
Yeah, so personally, I do genetic testing on everybody who comes in with ALS, but I would never do genetic testing on a family member without genetic counseling. That needs to be done. But the reason that I do it now even more enthusiastically than I used to is because now we have a drug for people with SOD1. I want to know whether you have SOD1, because if you do, I'm going to offer you this drug. So that's another very compelling reason to do it.
Good distinction. Mm-hmm. Is there any link between gut health and ALS?
Potentially. So this has been an area of interest. We did a study here. So yes, we've even collected poop. That's one biofluid we didn't talk about. But so what we know is there do seem to be gut changes that can be measured in people with ALS that look different than healthy controls. And so the gut actually has a very robust immune system and immune markers you can check. And so there's different patterns of inflammation that you see in ALS. And so the question that's not answered is, can you intervene on gut health or change the microbiome balance to change progression in ALS? And that's to be determined. But there are these changes that have been measured, and it is an area of research interest. So potentially, the research question that's still unanswered is, can you intervene on gut health and make microbiome changes to change progression in ALS? And that's still to be determined. But it is an area of active research, and something that's in this big database as well that's being looked at. And that's another one where AI might be a really good tool, because there's so many things you can measure in the microbiome. You might need some of these big fancy AI models to kind of sort that out.
Okay, so more to come on that as your research progresses. So we've had several questions about the C9 mutation. Can you explain what that is?
The C9 mutation is a mutation.
In layman's terms.
I'm sorry?
In layman's terms.
No, I'm going to get to that. It's actually a very interesting mutation. There are a bunch of neurological disease that are called repeat-associated gene mutations. Our genome, or all the DNA we have, which we call the genome, okay, has certain regions in it where there are certain patterns of the DNA that are repeated again and again and again and again. And this is normal. It happens many, many, many different genes, okay? But we have found over the last 50 years, really, that there are some diseases that are associated with too long a repeat. And some of those diseases include Huntington's disease, myotonic muscular dystrophy, Friedreich's ataxia. There's a bunch of, and in fact, Fragile X mental retardation in children. And so what was found in 2011 is that these were families that had ALS, but we couldn't find the gene, okay? We knew that there were multiple family members that had ALS, but couldn't find the gene mutation. And it turns out that this C9orf72 mutation, most of the ones I just told you about are called trinucleotide repeats, where you have the repeat is 3 bases, and then it goes 3, 3, 3, 3, 3, 3. This one is 6, 6, 6, 6, 6, 6, or a hexanucleotide repeat, okay? And that made it very difficult to find from a molecular biology level, because technically it's hard to find these 6 repeats. And so it was found back in 2011 by multiple groups that the gene, which is on chromosome 9, which gives it the C9 designation, okay? ORF, meaning open reading frame, okay, which means it is translated into a little protein, okay? 72, which is its marker on the gene, that's where it gets its name, C9orf72. That it had these hexanucleotide repeats. Now, most of us have hexanucleotide repeats in this same gene somewhere between 2 and 8 or 10. We all have repeats, okay? People with ALS or frontotemporal dementia have more than 30. And some of them have up to 1,000, okay? And so what we haven't figured out yet is, as opposed to some of these other diseases I told you about, like myotonic muscular dystrophy, the more repeats you have, the worse your disease is. Or in Huntington's disease, the more repeats you have, the worse your disease is. Turns out that's not the case here. That you can have 1,000 repeats or you can have 45 repeats, and your disease can be just as bad. So we haven't quite figured that one out. But it's a unique kind of mutation that causes pathology that we can follow and identify in human tissue. And that's what's so important about it. It creates these little proteins that we call dipeptide repeat proteins that we can actually measure in spinal fluid, we can measure it in brain, and it actually allows us to have, again, a biomarker of whether we've actually done anything or not done anything when we've tried to treat patients with this disease. So the data that I showed you up here, we looked at those little proteins that are made by C9, and they were unchanged in the patients who were treated, which told us that we didn't really do what we were hoping to do. So that's a little primer on what C9 is. If you have any other questions, I'm happy to.
How many people have C9? Like all of ALS?
In all of ALS, well, it's the most common mutation. The argument is that in people with familial disease, meaning that you have an inherited disease, your father had it, your mother had it, your cousin had it, so-and-so, about 50%, at least in the United States, are related to this C9 mutation, okay? And we've found that in our clinic. The other argument is that about 7% of people who have sporadic ALS may have a C9 mutation. And so we look for it in everybody.
Mm-hmm. Thank you. So we've had quite a few questions on research, and we have more questions to get through. This one's kind of a loaded question. How will cuts in NIH funding affect your ability to conduct research and advancement in treatments?
It will. It's been really tough. So I think the ALS community relies very heavily on federally funded research to advance this. And you know, just from a practical standpoint, like we're on a hiring freeze at Emory for research staff, right? So we might want to do a new trial, and Emory won't let us hire somebody to work on that trial because of the federal funding cuts like impacting universities. So Yes, it's very much, if you're prone to advocacy, I would think advocating that, federally funded research should not be a partisan issue, and it's something that we should support through all administrations is a really important thing to do. It's very much impacting ALS research.
It's really an attack on science. That's what it is. The idea that we can, I admit we can't cure ALS, not yet, someday, okay? But there are many diseases that we can cure. What's happening in the cancer world right now is extraordinary. There are people with cancer who would have died in 6 months who are now alive 5 and 10 years later because of the research that has been done in this country. And we are the leaders in the world with biomedical research. We are it. We have been, anyway. The attack on the NIH is, I don't understand it. If there's anything that the United States does really well, it's science. It's really a huge blow to the scientific community, and that will trickle down to the ALS community as well. The stuff we're doing is expensive. That's the reality. It's expensive. I can tell you that the proposal out there right now is to cut what's called the NINDS, or the National Institute of Neurological Disease and Stroke, which funds most of the ALS research, in half. Cut it by half. That's billions of dollars. I'll stop.
So what I'll add with that is we at ALS United of Georgia, along with other colleague ALS organizations across the country, do advocate for federal funding and for funding at the NIH and the Department of Defense that work specifically for ALS research projects. And we want you all to participate with us. And the way you can do that is on our website, there's an advocate tab that we are building out stronger, but you can sign up to be an advocate, and that just means when there's a call to action, when we want you to reach out to your senators and House representatives to say, hey, don't do this, this is why, this is personal to me, we'll help you have that email and send that out. The more they hear from their constituents, the more it helps. And so we are active on that front, and will continue to be so. So join us. Okay, we're going to talk a little bit about medications. So the first question is, are there medications or therapies being used outside the U.S. that have potential here? Maybe aren't here yet. That's
So
kind of broad, I know.
yeah, without going into specific medications, there are, anytime a drug is considered for approval, it has to be approved by each country's governmental agency, right? So the FDA approves drugs here, the European agency approves drugs, and each country has its own process. So there is a bit of variability when a drug comes through, particularly if it's a drug that is of questionable benefit, right? Like it might have a modest effect, it might not. The trial results were mixed. There are cases where drugs then will get some approvals and not get others. So I think if there's a game-changing drug, everybody's going to approve it. But when in these cases where the data is squishy, there's some countries approve it and some don't. And we've seen some variability. Like for example, the Amylyx drug that was approved and then a bigger trial showed it didn't work and it was pulled. Like some countries never approved that drug in the first place, but the FDA did. So yes, there's some variability. No, there's not any game-changing drug out there that's approved elsewhere that's not approved here.
Okay, I like that word, squishy. That's good.
Very
descriptive
scientific term,
there.
yeah.
Are there other medications besides riluzole for slowing the progression of ALS? And if so, and what are the differences in those meds? That might, I don't know.
I'll take it if you want.
Yeah, my answer is no.
Mine is tofersen, which is only for a subset.
Yes, I'm sorry, tofersen, yes, for people with SOD1. But the only drug that's ever been shown to slow disease in ALS and prolong survival is riluzole.
Because there's probably questions. So Edaravone is FDA approved. The backstory with that is that the trials have been sort of a mixed bag. So there was a trial in kind of a broader ALS population that showed that there wasn't a benefit. So then they looked at a subset and did a trial in that subset, and they showed some modest benefit. And then they did a larger phase 3 study in Europe, which did not show a benefit. So many of us think that the drug probably is not much better than placebo, but it is approved based on that one trial, and the safety data is good. So there's some people who choose to take it, and many who don't. The only other one that I think, the high dose B12 is one that has been, there is some non-US based trials that show that maybe if you give it very early on, that there's a very modest benefit. So that is out there as well. So none of these are game-changing things, but I think things that you'll see, and that's why I bring them up.
Are there any new therapies or medications for treatment and management of excessive saliva, which is the symptom?
I don't know that there's anything new, but there are multiple options. There's several medications that dry up saliva. There's Botox injections, which are given in the salivary glands to reduce production. There's things like suctioning devices and things that can help. So there's different strategies. It's not a one-size-fits-all, so that's kind of a talk to your doc about what's best for you sort of advice.
Okay.
But I don't think anything that's been really newly approved for that.
Okay. Is there any evidence, or what are your thoughts on medical marijuana helping with anxiety for ALS patients?
I will say we don't have good data on this because there's really not like robust clinical trials on marijuana use. So I will tell you this, that many patients try it. Anecdotally, I've heard some people say they find it helpful for certain symptoms. The ones that I've heard anecdotally, maybe appetite, some people find it helps with mood. But we don't have good clinical trials on this. So we don't have any really overt safety concerns. So it's kind of one of those things, if people want to try, we don't object to that. The Georgia process, because we get asked about this, so there is a Georgia THC registry. So in the state of Georgia, it's only like 5% THC oil is approved for certain conditions, and ALS is one of those. So if people want to pursue that, I think we put you on the registry, you pay the Department of Public Health, I think $20, so you get a card, and then you can get this THC oil. It doesn't apply to other forms of marijuana. But it really, all we have is kind of anecdotal information. It hasn't been studied in like these big clinical trials.
No real game changer there then.
No, I think we don't expect it to like dramatically change the course of ALS. Mm-hmm. Some people might find it helpful for symptoms, but we don't think it's like a specific ALS treatment.
Right. Okay. This one's a little more specific. What is CRISPR, CRISPR, clustered, regularly interspaced, short palindromic repeats?
Let me take that one.
Go for it.
It's really cool. I just got to tell you that. This is something that was discovered in bacteria. It's the way the bacteria deal with actually bacteriophages or viruses that can infect them. And I don't know, did she get the Nobel Prize yet for this? I'm not sure. But anyway, CRISPR is a way of gene editing, okay? So what I was telling you about with ASOs, this is called gene silencing, turning genes off. Gene editing means you're taking a piece of a gene, usually a mutation in a gene that may cause a disease, and you're going in and you're cutting it out, and taking it out, and then you're replacing it with the normal sequence. So a mutation is an abnormal sequence of DNA. You're going in there and you're taking it out and you're putting a new one in. It's remarkable. Bacteria learned how to do this billions of years ago, I'm sure. And we have now discovered it, or these very smart scientists have discovered it, and it is now being brought to the clinic. It was science fiction just a few years ago, but it is now being used to treat genetically based diseases. There's an extraordinary example of a young boy in Pennsylvania, at UPenn, University of Pennsylvania, who was born with a disease, I can't even remember what it was, but a genetic mutation that basically he was going to die from pretty quickly. They very quickly, this was in the newspapers, it was in the New England Journal of Medicine, very quickly created a CRISPR, which is a combination of basically what we call a guide RNA, and which finds the DNA sample that is abnormal, goes in there and clips it out, and replacement DNA, and basically created this combination. It's called CRISPR-Cas9, and cured this kid. Unbelievable. It's like, wow. And in fact, there is some examples in sickle cell disease now of people being cured with CRISPR-Cas9. So here's this experimental thing that was found not that long ago. It was really a laboratory phenomenon that has now come to human medicine. In fact, at our NEALS meeting, the Northeast ALS Consortium, that will happen in October, I am leading a presentation about gene therapies for ALS, and we have the person from UPenn who did this CRISPR-Cas9 on this young kid who's going to speak to us about ALS. Can this be done for people with mutations with ALS? So watch this space, because I would have told you just a few years ago, no way this is going to happen. It's going to happen. There are some really smart people at Stanford and at UCLA who are working on this. I've seen some of their data. It's extraordinary. So again, it's going to be specifically for people with gene mutations, but it's, again, another technology that has come from basic science, from the laboratory, from NIH sponsored and NIH funded research, which is now hopefully going to make it to the clinic at some day. It already made it for some diseases. It's, I think, going to make it for ALS in the next several years.
Okay. I'm not sure if I'm pronouncing this right. It says, have you received info on ibogaine treatments and its improvements on rebuilding neural pathways? Is that? I don't know much about it. That's a fair answer.
Yeah, I don't know much about it. I've looked it up. I haven't seen any really good data on it, but it's out there. I don't know much about it. The only thing I found is some stuff on the web.
Okay. So maybe more to come later. Is there any research surrounding low microdoses of psychedelic treatments?
I have seen that there's some studies on psilocybin and ALS. So it is something that's being studied, I think more for like quality of life and improving kind of mental health outcomes in ALS. But I haven't seen final data on it, but it is something that's being studied. So it's more to come on that one as well.
Yeah, there's a big program going on at Hopkins now on that. Yeah.
Okay.
I know we got a
We have
psychedelic and marijuana crowd here, huh?
I'm very impressed by the questions, a whole wide range here. We have about 5 more questions that we've captured so far. So if you have a few more out there that we haven't captured, let us know, because we might have time. But so just to kind of keep going on those few that we have left, are there any stats on the percentage of veterans that have ALS in Georgia and nationally?
There are. I don't have them memorized, but I can tell you that, so ALS does happen a bit more frequently in veterans compared to civilians. So I think where the number is something like 2 out of 100,000 civilians get ALS.
We
and
quote twice as many veterans as civilians.
3 to 4 out of 100,000 veterans will get ALS. So it is more common. We don't know the reason for that. It is an area of active research as well. So but I don't think I have numbers beyond that.
Okay. Well, in addition to the stats on veterans with diagnosis of ALS, do we know if there's any connection to burn pits in military service or hazmat exposure? Have you seen any research on that?
So we haven't found kind of what about military service is the cause of increased risk. They've looked at head injury, they've looked at specific tours of duty in certain service locations and branches of the military, and there hasn't been like, this is the thing. So it is an unanswered question that we should answer, but we don't know yet.
Right. Can PLS change to ALS diagnosis?
Yes. So PLS is primary lateral sclerosis. So this is where you have the increase in tone or the spasticity and brisk reflexes and changes to the speech and limb strength without the muscle wasting and the twitching and that sort of thing. And so when somebody first presents with what looks like PLS, it can stay like that, where they only have these what we call upper motor neuron signs through the course of things. And some people present where it looks like PLS, and then they later develop the signs of classic ALS. So it can change with time, and that's something we monitor for. And actually a study that we participated in, which is if you see somebody on day one that looks like PLS, are there biomarkers or clinical signs that will predict progression? So we're looking at the answer to that question as well.
Okay. How do we donate brains to the brain bank, and can families learn from these findings? You touched a little bit about that early in your presentation.
So we do accept brains and spinal cords from our patients that we have taken care of, and the important thing about that is we have all the clinical data on them. And so just an autopsy without clinical data is not terribly, it's not as useful as having all that longitudinal clinical data associated with it. The answer is yes. If you're interested in it, we can talk to you about that, especially if you're one of our patients, and we can figure out the arrangements to do that. I can tell you that it's expensive, and we are right now having to pick and choose because of some of the money that we don't have anymore. We will take all the autopsies that are offered to us as long as we have that ability. And so if you're interested, get in touch. You can talk to Jen or Priya over there and let us know.
Okay. Couple more. Have you identified any somatic mutations that could be potential drug targets for sporadic ALS patients?
No. It's interesting. When you talk about a somatic mutation, it's a mutation that occurs after you're born. So we all have DNA that we were born with. And those mutations are, all the mutations that we know about for the most part, are present in our genome from the time we're born. A somatic mutation is the idea that your gene is somehow mutated, probably by some environmental factor, during your lifetime, after you're born. There are some questions about some mutations where there's no family history, whether that's a somatic mutation, but it's never been proven. So as far as I know, the answer to that is no, there are no somatic mutations that we know about in ALS.
Okay, one more specific question. In a case reported this week, an SOD1 positive patient on tofersen reported by EMG, her disease had not progressed since the start of therapy. Does this mean that she had a single pathogenic pathway from normal to ALS, and the other patients have single or multiple pathways? And what are the implications for personalized or general therapeutic approaches? I know that's a lot.
Well, yeah, so there are reports of people on tofersen who have an SOD1 mutation that stop getting worse, and there's actually some reports, I don't think I've seen it yet, maybe Christina, you are convinced, but I'm not sure I've been convinced that anybody's getting better, at least in our group, but that has been reported by several groups, reliable groups, so I believe it, okay? It doesn't work in everybody, and that's a real question. So we have had patients, at least in our original trial for tofersen, who got the highest dose, who still died. So it doesn't work for everybody. And so your question of whether this is what we call a monogenic gene mutation that just causes the disease, I don't think that's answerable with the tofersen. I can tell you, we have a research program that's going on based on identical twins who have an SOD1 mutation, but they look very different, but they're identical twins. So there are other genes probably that drive disease in people with SOD1. But right now, as far as we're concerned, SOD1 is a disease-causing gene that can be modified with tofersen, but not in everybody.
Okay. Okay, my last two questions, and then if you all did not get a chance to talk to one or some of our community partners, they will still be here for a short period of time when we dismiss. So I wanted to encourage you, don't forget to check them out and say hello to them. So are there plans in place? I'm sorry. And don't forget to do your evaluations on the table after our last two questions. So are there plans in place for phase 3 testing of ALS therapy in P001?
I think they are planning now a phase. So this was a anti-inflammatory drug that had a phase 2 study that was negative, but then they looked at a subset and said maybe it works in the subset, and then the company was bought by a different company, and now that company, I think, is planning a phase 3, but I don't have any official information on it. I think probably the press thing you read is the same thing that I read, so I don't have any insider tips yet. So it's an anti-inflammatory drug. It's actually IV bleach. And what they tried to do was identify patients that had high inflammatory markers and target those patients, but I don't think we knew the best way to do that. And so it's really important to note that eating bleach is very toxic and not safe, so don't do that.
It also doesn't treat COVID.
So we all know that there is not one definitive test that gives us a diagnosis of ALS, but has it become less of a disease of exclusion over time or become quicker or easier to diagnose?
This is a tough question for me because it frustrates me terrible. I don't think this is a disease of exclusion. It's a very easy disease to make a diagnosis of. You just got to know what you're looking at. And I tell my students and my fellows, there is no differential diagnosis. When somebody comes in who clearly has ALS, it's ALS. Now, if you ask me what is ALS, I'll tell you I don't know, because I just don't. I mean, I can identify it, I can tell you what it is, I can tell you what's going to happen, but I don't know what it is. And that's the whole point of what we're doing in the laboratory. What is driving this disease? Why do people get it? But this is not a disease of exclusion. All these MRI scans that are being done, all these surgeries that are being done, all these things, they don't need to be done. As long as the physician who's looking at you knows what they're looking at. That's why we have really, and Dr. Fournier has really pushed this, we have something called the RACE clinic, or the Rapid Assessment.
Rapid ALS clinical evaluation.
Rapid ALS clinical evaluation. We have begged clinicians in the community, if anybody comes in with any of these kinds of symptoms, just send them to us. We'll tell you whether it's ALS or not. Stop doing all this other stuff. I think I'll stop there.
Okay. Well, we are so appreciative of your time outside of the patient room today. I know this is extra time for you all to take to speak to this community. I guess just in wrapping up, I would love for you all to just share, I mean, obviously, the amount of time and work you put into this has become your life's work, but what kind of drives that passion behind it? You know, the ultimate goal for each of you in this work. I think I know the answer, but if you want to just kind of wrap it up a little bit with any final words, it would be great.
Yeah, I mean, I don't think we have to tell you how important this is, right? I mean, we see all of you, and the call to action is incredibly important, right? We have to come up with treatments for this. We don't want people in 5 years and 10 years to have to go through the same process that the people in this room have been through. And so this is what gets us out of bed every day doing what we do, is it's super important to take care of everybody that has ALS today, and it's equally important to make it better for people who might go through this in the future. So I think we're all on the same team here of how important this work is. And I think especially given what we've seen happen with ALS research, and because I hear this in the clinic sometimes of like, we've known about this since Lou Gehrig, and nothing's happened. And I know it feels like that, but I think in the lab, we feel like things are happening. And that I think in my career, we're going to have better treatments for ALS. And so that's what gets me out of bed every morning to do this.
Well, and that's our goal, to make sure what's in the lab comes to light, too, right? As partners with the Emory ALS Clinic and ALS United of Georgia, working together to serve you all, but also to bring that to light and let you know what is happening in Georgia and nationally. I mean, these are worldwide researchers here that you have in our own backyard, and we're just so proud to be able to stand next to you and be able to bring this to everyone today. So thank you so much.